Good evening, friends and colleagues. I am Professor Barend Erasmus, the Dean of the Faculty of Natural and Agricultural Sciences at the University of Pretoria. This evening, it gives me great pleasure to welcome you to the inaugural address of Professor Anabella Gaspar from the Department of Biochemistry, Genetics and Microbiology at the University of Pretoria. An inaugural address is a benchmark event, and I really look forward to what Professor Gaspar will share with us on antimicrobial drug discovery. For a brief introduction and a brief welcome of Professor Gaspar, I hand over to my colleague, Professor Themba Musia, the Vice Principal Student Life at the University of Pretoria.
Thank you, Mr. Dean. Good evening, ladies and gentlemen. Welcome to this special occasion at our esteemed University of Pretoria. And I hope we'll enjoy this evening with Professor Anabella Gaspar as she keeps us glued on our screens with her exciting talk. On behalf of the Senate of the University of Pretoria, I would like to take this opportunity to extend a special word of welcome to Prof Gaspar herself, her family, her friends, her colleagues in the Department of Biochemistry, Genetics and Microbiology in this country and elsewhere, and of course colleagues in the executive, the dean and the deputy deans, and all the senior and other members of staff in the faculty and the university. And I would also like to thank our students for joining us this evening. I mean, we're glad that we will be listening to this this evening, and this kind of inauguration is a very special one. But in the life of an academic, graduation is one of those very first special occasions that you have, and of course, these graduations are usually followed by multiple graduation ceremonies, and we celebrate, and then we also have excellence academic awards in which we celebrate the academic achievements of colleagues. But this particular one is special in its own way to actually have an inaugural address where a scholar, a full professor in this case, or a person appointed in a senior position in the academic leadership will share with us their distinctive discoveries. And it is at this, in our tradition as universities, that we sit down and enjoy and listen and learn as we move forward. And I'm sure this evening she will speak to us about biochemistry and the development of microbial drugs. But first, who is she? Professor Gaspar is a principal investigator of Biotherapeutics Research Group. She's also a chair of the Division of Biochemistry, a position she held since 2018, and was promoted to full professorship in 2021. She is also a member of the European Peptide Society and has served as a council member of the South African Society of Biochemistry and Molecular Biology. Well, she's always been an achiever, really, since 1982 and 1985, and all her degrees has been cum laude. She received her first degree, a BSc in biochemistry and chemistry, followed by honors and a master's. As I said, all of them cum laude, so that attests to her level of achievement in academia. And later, of course, she earned her PhD in 1995 from our university. And in 1997, she was promoted to senior lecturer. And it goes on and on. And today we're celebrating her full professorship, and she will be able to share with us, what is happening in biochemistry. I'm told it is, the study of chemistry of life, what goes on at molecular level biochemists tell us as living organisms, the kinds of things and complications that happen there. And she is an established researcher in biochemistry, and she's particularly involved in the field of biotherapeutics, and her research concerns the discovery of microbial and anti-diabetic peptides, understanding the structure, activity, relationships, and modes of action with these peptides. And this is very necessary for novel therapeutics. At present, her main research focus is the discovery and development of antimicrobial peptides, AMPs, as they are called, to address the problem of antimicrobial drug resistance. A UK SAMRC Newton-funded project, which lasts until 2023, focuses primarily on the development of peptide-based antibiotics targeting gram-negative resistant pathogens. And the main aim of this project is to establish a program that centers on heat-to-lead development of AMP therapeutics in South Africa. While her research keeps her busy, she's also involved in teaching at both undergrad and then postgrad. And I guess this is also one of her passions, particularly undergraduate teaching. Now, she has published 30 articles, research articles, peer-reviewed, and she has numerous other national, and international conferences where she participated and shared knowledge. She has co-supervised 48 honors students, 24 MSCs, and of course, we also see that she has done supervision for 5 PhD candidates. And she's still busy with 2 more honors, 1 master's, and 5 doctoral students. Because biochemistry is continuously growing, new knowledge and technologies continue to change our understanding of the discipline. This necessitates that the curriculum be reviewed and updated regularly. But Prof Gaspar will share with us tonight in her address titled From Basic Biochemistry Concepts to Antimicrobial Drug Discovery. And during this lecture, she will present an overview of her research that has been continuing and developing these peptide-based antimicrobial drugs against bacterial and yeast infections, and more recently targeting resistant gram-negative infections. She will also show the link between basic biochemistry concepts and antimicrobial drug discovery, and highlight the importance of multidisciplinary international collaboration for capacity building in peptide-based microbial drug discovery. And of course, training of the next generation of scientists. And given our recent context of the pandemic wreaking havoc, we are particularly interested to hear what groundbreaking information she will share with us in her research. So without any further ado, ladies and gentlemen, I present to you our esteemed colleague, Professor Anabella Gaspar, and I trust that we will all enjoy her presentation this evening, and I thank you.
Professor Musia, Vice Principal Student Life at the University of Pretoria, Professor Erasmus, Dean of the Faculty of Natural and Agricultural Sciences, Professor Naidu, Head of the Department of Biochemistry, Genetics and Microbiology, colleagues, students, family and friends, good evening. I am extremely grateful for the opportunity to present this inaugural lecture, From Basic Biochemistry Concepts to Antimicrobial Drug Discovery. In the first biochemistry lecture that I attended as a second-year BSc student in 1980, the late Professor Daniel Potgieter wrote the word biochemistry on the blackboard and explained with a quick calculation that biochemistry was a combination of 1/4 biology and 3/4 chemistry. The textbook prescribed at the time was the 4th edition of Outlines of Biochemistry by Connan Stumpf, which divided biochemistry into 3 sections: chemistry, intermediary metabolism, and a smaller 3rd section on informational molecules. The main emphasis then was for students to understand the basic chemistry principles underlying biochemistry. And at the time, biochemistry in the final year of the 3-year BSc program could only be combined with chemistry. Fast forward 40 years, and biochemistry has evolved into a multidisciplinary science. One of the textbooks prescribed currently for the BSc Biochemistry program, Principles of Biochemistry by Leninger, divides the content into 3 main themes: structure and catalysis, bioenergetics and metabolism, and information pathways. The emphasis is no longer only on building biochemistry on basic chemistry principles, but also on cellular, physical, genetic, and evolutionary foundations. Biochemistry also provides the foundation for other science disciplines and is no longer only combined with chemistry. Biochemistry is continuously growing, and the emergence of new knowledge and technologies continue to change our understanding of the discipline. Although this necessitates that the curriculum be reviewed and updated regularly, a solid foundation of basic biochemistry concepts is central to the understanding of life at the molecular level. One of the key concepts in biochemistry is the relationship between structure and function of macromolecules and the role of weak, non-covalent or molecular interactions relating to structure and function. Not only do molecular interactions play a role in the folding of macromolecules into active conformations, formation of supramolecular structures and organelles, but the binding of an enzyme to its substrate, a hormone or a neurotransmitter to its cellular receptor, a specific antibody to an antigen, a virus to a host cell, a drug to its target, are all as a result of molecular interactions. Moreover, a solid foundation of the fundamental principles behind the basic and more advanced techniques used in biochemistry is also essential. In a research-intensive university, teaching in biochemistry must continue to be combined with research, starting at the undergraduate level, to produce graduates with the necessary skills to address complex biological questions. My research career started as a PhD student when I joined the tick research group headed by Professor Albert Knights. I was fascinated when I became aware that tick salivary glands are rich sources of bioactive molecules. The focus of the research group was the characterization of tick salivary gland antihemostatics in search of potential anti-tick vaccine candidates. For my PhD, I biochemically characterized the first blood coagulation protease inhibitor, a factor 10A inhibitor, from the salivary glands of Ornithodorus savigny ticks. In subsequent years, the group identified additional antihemostatics and 2 defensin isoforms. From the outset, my main interest was whether tick bioactive molecules could be exploited for therapeutic development. It was then in 2011 that I started the Biotherapeutics research group with Professor Megan Bester, also a biochemist, at the Faculty of Health Sciences as co-investigator. Although recently we have also worked on anti-diabetic peptides, research conducted by Dr. Awal Ibrahim, a postdoctoral fellow, the main research focus of the group is the discovery of antimicrobial peptides, AMPs, to address the problem of antimicrobial resistance. Antimicrobial resistance, or AMR, is a threat to modern medicine, impacting both human and animal health. According to the World Health Organization, antibiotics are becoming increasingly ineffective as drug resistance spreads globally, leading to more difficult to treat infections and death. There is thus an urgent need to develop novel anti-infective drugs. In line with the World Health Organization list of priority pathogens, these drugs should be focused on gram-negative pathogens, while recognizing that gram-positive species and fungi continue to generate a significant health burden. Synthetic peptides, based on the sequences of naturally occurring AMPs, are promising candidates for developing novel antimicrobial drugs. AMPs are key components of the innate immune system of all living organisms. They are small polypeptides, typically made up of 10 to 50 amino acids. Most have a net positive charge, ranging from +2 to +11, contain a significant portion of hydrophobic amino acids, and are amphipathic. Their secondary structure varies greatly, and AMPs can adopt alpha helical, beta stranded, extended, or random coiled structures, or combinations of these structures. The mode of action for most AMPs is the disruption of membrane integrity of the target organism and/or by translocating across the membrane to affect intracellular targets. In order to disrupt or cross the microbial membrane, cationic AMPs first interact with the negatively charged phospholipid head groups of membranes. Once in contact with the microbial surface, the hydrophobic residues insert into the lipid bilayer, thereby destabilizing the membrane. Various models have been proposed to explain their membrane disrupting activity. AMPs have numerous attractive properties, including broad spectrum activity, rapid onset of killing, bactericidal and/or immunomodulatory activities, biofilm disruption, and a low incidence of resistance development. The primary challenges associated with AMPs are their susceptibility to proteolytic degradation, cytotoxicity of some AMPs to eukaryotic cells, reduced efficacy under physiological conditions, and high production costs. These limitations can, however, be overcome by designing shorter analogues and structure modification. In the next part of this lecture, I will present an overview of some of our research on tick antimicrobial peptides. Various shorter and modified synthetic peptides derived from tick defensin isoform 2, OSDEF2, a 37 amino acid peptide with a net charge of +6, have been investigated for antibacterial and antifungal activities. In this presentation, I will focus mainly on peptide OS, and analogues derived from OS, namely OS minus C, and non-amidated or amidated OS 3 to 12 and OS 11 to 22. To investigate their antibacterial potential, the structural and functional properties of the 22 amino acid synthetic peptide OS derived from the C terminus of the parent defensin was compared to that of a shorter analog, OS minus C, in which the 3 cysteine residues were omitted. Although shorter than the parent defensin, both analogues still carry a net charge of +6. Both peptides were bactericidal to gram-positive and negative bacteria, whereas the parent defensin peptide was only found to be active against gram-positive bacteria. This study showed that overall, OS and OS-C were more effective against gram-negative bacteria with rapid killing of gram-negative Escherichia coli. Scanning electron microscopy was used to investigate the direct effects of these peptides on bacteria morphology. In contrast to melittin, an antimicrobial peptide from bee venom, which showed distinct blebbing of both gram-negative and positive bacteria surfaces, cells treated with both peptides showed collapse of cell structures, indentation of cell membranes, and cell lysis. Furthermore, transmission electron microscopy studies revealed that gram-negative and positive bacteria treated with low peptide concentrations had intact membranes with distinct indentations, and the cytoplasm was pulled away from the cell envelope. Both peptides also adversely affected intracellular structures, causing different degrees of granulation of the intracellular contents. The ruffled membrane appearance indicated that the peptides may insert into plasma membranes, causing membrane destabilization and leakage of cell contents. To further investigate their mode of action, liposome leakage and confocal fluorescence microscopy studies were undertaken. Liposome leakage assays using a gram-negative model membrane showed that OS causes membrane disruption, whereas OS-C was less active, suggesting additional intracellular targets for OS-C. To determine whether these peptides are able to translocate across the membrane, cells were incubated with fluorescently labeled peptides, which fluoresce green. Confocal fluorescence microscopy showed that the labeled peptides were able to enter gram-negative and positive cells and accumulate intracellularly. Subsequent molecular interactions may thus compromise cellular function and morphology, as was observed with electron microscopy. In a further study, even shorter peptide analogues of OS were evaluated for antibacterial activity. The purpose of this was to understand the structural requirements for activity. Out of 8 partially overlapping sequences, only the 10 residue OS 3 to 12 and the 12 residue OS 11 to 22 analogues were active when screened against gram-positive and negative bacteria. The observed reduced activity may be due to the decrease in the net positive charge of +3 for the shorter peptides versus +6 for OS. This also highlights the importance of both the N and C terminus regions for activity. Secondary structure is an important feature of AMPs required for the insertion and destabilization of bacterial membranes. The reduced activity of the shorter peptides compared to OS may also be as a result of the different structures adopted by each, since far UV circular dichroism, CD, data showed OS to have some alpha helical properties, whereas in contrast, the shorter peptides remained coiled, despite incubation in SDS, a membrane mimicking environment. In an attempt to increase the antibacterial potency, the two shorter peptides were carboxyamidated. This increases the net charge of the peptides by +1, as well as their resistance to exopeptidases. In general, the activity of the amidated peptides against gram-negative and positive bacteria, except for gram-positive Staphylococcus aureus, was several-fold greater than for the non-amidated analogues. The enhanced activity of the amidated analogues is most likely due to the increase in net positive charge, resulting in an increase in the electrostatic attraction between the peptides and the bacteria membranes, as far UV-CD analysis showed no differences in the secondary structures of the non-amidated and amidated analogues. Time-kill assays at their respective minimum bactericidal concentrations showed more rapid killing kinetics for the amidated 3 to 12 peptide against gram-negative E. coli than gram-positive Bacillus subtilis, whereas the amidated 11 to 22 peptide took longer to eradicate gram-negative and positive bacteria. Liposome leakage assays, again using a gram-negative model membrane, identified the amidated 3 to 12 analog also as membrane disrupting, while the amidated 11 to 22 peptide was less active, suggesting, in addition to membrane permeabilization, other targets may be involved. An important finding was that the membrane disrupting effect of the amidated 3 to 12 analog was not diminished in the presence of salt, identifying it as a promising candidate for further development. As AMPs have broad spectrum activity, the tick-derived peptides were also evaluated for antifungal activity using Candida albicans as a model organism. Various analogues have been investigated for their antibiofilm capabilities, but for the purpose of this presentation, I will only present their effects on planktonic cells. OS, OS-C, and the amidated 11 to 22 analog were the most active, with minimum fungicidal concentrations in the micromolar range. Candida albicans killing occurred within 30 minutes for OS and OS-C, whereas the amidated shorter analog took longer. Both OS and the amidated analog caused leakage of fungal liposomes, indicating membrane disruption. In contrast, OS-C had a lesser effect. Confocal fluorescence microscopy studies showed that all three peptides were able to translocate into the cytosol of Candida cells. To investigate whether peptide translocation is an ATP-dependent process, translocation was performed in the presence of sodium azide, an inhibitor of ATP synthesis. Under these conditions, OS and OS-C were no longer able to enter cells, suggesting that for these peptides, translocation into the cytosol is an energy-dependent process. In contrast, uptake of the amidated 11 to 22 analog was unaffected by ATP depletion, and although at a lower intensity, the peptide was still present in the cytoplasm. Translocation of the peptides into Candida cells therefore suggested that they may affect intracellular processes. to investigate whether these peptides target intracellular membranes or membranous organelles such as vacuoles, peptide translocation was performed using Cell Tracker Blue, a vacuole-specific dye. Flow cytometry and confocal fluorescence microscopy showed that at higher concentrations, only OS caused significant vacuole degradation, suggesting that OS has a direct membrane targeting effect, while for OS-C, cell death is due to a more indirect mode of action, such as reactive oxygen species or ROS generation. The formation of intracellular ROS has been proposed to play a key role in the fungicidal activity of several AMPs. Although all three peptides induce the production of ROS, co-incubation with ascorbic acid, an antioxidant, revealed that only ROS generated by OS-C, and to a lesser extent, the amidated 11-22 analog, resulted in cell death. However, for therapeutic application, AMPs need to effectively kill microbes while showing absence of toxicity towards mammalian cells. The various tick AMPs display negligible cytotoxicity when tested against erythrocytes, leukocytes, and different mammalian cell lines. An attractive feature of the tick AMPs is that they are multifunctional and have bioactivity beyond microbial killing. Other novel properties that we have identified are antioxidant, anti-endotoxin, and anti-inflammatory activities. Although these studies showed that these AMPs are promising candidates for further development, additional modifications are required to increase their potency, stability, and activity under physiological conditions. Since January 2021, the group's main research focus in collaboration with our UK partners at King's College London, Public Health England, and the Universities of Warwick and Sheffield is the development of peptide-based antibiotics targeting gram-negative resistant pathogens. This project is one of two projects funded by the South Africa-UK Antibiotic Accelerated Drug Discovery Programme, an initiative of the South African Medical Research Council and the UK Medical Research Council. For this project, we have identified a panel of 45 AMPs from various African species, namely tick, frog, scorpion, and primate, which best reflects biological diversity on the continent. The main aim of this project is the establishment of a program focusing on hit-to-lead development of AMP therapeutics at the University of Pretoria. This will be achieved by screening the initial panel of AMPs and the rationally designed second-generation peptide analogues using in vitro and ex vivo assays designed to better replicate specific infection settings. This will be followed by evaluation of the lead peptide candidates for cytotoxicity and stability, in vivo toxicity, and efficacy. Different aspects of this project will also be supported by the Drug Discovery and Development Centre, H3D, University of Cape Town, and the Antibiotic Accelerator Initiative Drug Discovery Partnership Hub at Rhodes University. Through this collaboration, 3 PhD students and 1 early career academic will be trained in advanced AMR technologies in the UK, and skills will be translated back to UP. For rational drug design, training in various biophysical techniques, and molecular dynamics, MD simulation, of peptide interactions will take place at King's College, London. Training in the use of porcine ex vivo models of cornea bacterial keratitis and chronic lung infections will take place at the universities of Sheffield and Warwick, respectively. Both ex vivo models mimic in vivo biofilm infections and thus can more accurately predict in vivo success of lead AMP candidates. Due to COVID lockdown and travel restrictions, the PhD students were instead trained online in MD simulations to evaluate in silico the interaction of 4 tick amps with models of gram-positive and negative membranes. MD simulations are the key to a molecular level understanding of the role of each amino acid in AMP activity and thus their overall mechanism of action. The simulation that you just saw shows how the AMP OS interacts with a gram-negative model membrane. Further analysis of the interactions of OS and OS-C with a gram-negative membrane indicated that upon interaction with the model membrane, the secondary structures of these peptides differ considerably. OS adopts a beta sheet conformation, whereas OS-C adopts a random coil turn structure with both peptides existing as monomers and dimers. Furthermore, both peptides form extensive hydrogen bonding with the lipid head groups of the membrane bilayer, involving specifically their N-terminus lysine 1 and arginine 4 residues. The lysine residues in the C-terminus of the peptides also form hydrogen bonds with the lipid head groups. The initial electrostatic interaction of the peptides with the gram-negative membrane bilayer may be seeded by these positively charged residues and play an important role in peptide membrane interaction. Also, the OS peptide was found not to insert into the gram-negative membrane, but localize at the water membrane interface. In contrast, OS-C inserted into the membrane via the N-terminus residues. Currently, the acquired MD simulation skills are being applied by the 3 PhD students to evaluate the interaction of peptide hits identified after the first round of screening with model membranes. This information will aid in the rational design of second-generation peptides with increased gram-negative targeting activity. In conclusion, through this research, I have become more aware of the importance of the basic concepts taught in the biochemistry curriculum, as these are key for the development of new therapeutics. Understanding the structure-function relationship of bioactive molecules and the molecular interactions between molecules and their targets are essential for the rational design of new therapeutics. It is thus essential, important, that teaching of the basic biochemistry concepts continue to be linked with research or other relevant applications to reinforce students' mastery of key concepts required for understanding life at the molecular level. Furthermore, multidisciplinary, national and international collaboration are necessary for capacity building in peptide-based antibiotic drug discovery and the training of the next generation of scientists in advanced AMR state-of-the-art technologies. I believe that through these advanced technologies, the challenges associated with AMPs will be overcome and ultimately deliver lead AMP candidates from Africa's rich biodiversity to fight AMR. closing, I would like to acknowledge the University of Pretoria for allowing me to live my passion. A special thank you to both past and present colleagues for their support and friendship throughout my academic career. I would also like to thank past members and postgraduate students of the TIC research group for their contribution to the field of TIC bioactive molecules. In particular, I would like to acknowledge a very special person, Professor Albert Knights, for his excellent mentorship, guidance, wisdom, and words of encouragement over many years. I would also like to thank Professor Knights for his co-supervision of a number of postgraduate students working on TIC AMPs. I also want to acknowledge Professor Megan Bester, Dr. Elena Tauta, and Dr. June Serum, co-investigators of the Biotherapeutics group, for their support and commitment. In particular, I would like to acknowledge Professor Megan Bester for her substantial contribution to TIC AMP research over the years, and more recently with the management of the AMR Drug Discovery Project. We make a good team. I would also like to thank all of my postgraduate students, both past and present, for their enthusiasm, dedication, and their contributions to my research, some of which I've discussed in this presentation. I would also like to acknowledge my local and international collaborators for their substantial contributions, as well as the funders of my research over the years. And finally, I would like to thank my Heavenly Father for always holding my hand and blessing me with a wonderful family. I would like to acknowledge my late mother, who had no idea what biochemistry was all about, but made sure to inform everyone she met that her daughter was a biochemist. I would also like to say a very special thank you to my father, husband Philip, daughter Katleya, and son-in-law Richard for their love, encouragement, and continued support. And of course, my grandson Angelo for all his love. Thank you.
Thank you very much, Professor Gaspar. In the last few months within the faculty, we've had a series of expert lectures, and one of them was on the value and of African biodiversity. There was an oversight that we did not feature this wonderful work from Professor Gaspar. Who would have thought that some of the new therapeutics in the future might rely on tick saliva coming from somewhere on the African continent? What a wonderful and interesting story, and I wish you well with the rest of your career and what is obviously very promising research. We'll be watching closely because I see enormous potential here. I also appreciated the way in which you linked the teaching and research. Within higher education institutions, there's always a debate on the links between teaching and research, how the one feeds into the other. The consensus, though, that does exist is that without excellent researchers like Professor Gaspar, we would not have excellent teaching. And this is something that she hasn't mentioned, but that is also something that she does, excellent teaching. Professor Gaspar was instrumental in the move to hybrid learning within her department, and she's well known within her department, within the faculty, and also in the university for substantial contributions and innovation in teaching and learning, always putting students first. For that, I'm deeply thankful for this contribution that you make to the faculty's core business. There's another element that I'd like to thank her for, and this is for the leadership within the department. Some time ago, the current Department of Biochemistry, Genetics and Microbiology was the end result of an amalgamation of individual departments. One of the resulting divisions, that of biochemistry, is now under the leadership of Professor Gaspar, and for that I thank you. I'm under no illusion of how much additional work and additional input is required from you, not to only keep your own research going, not only to teach at the level at which you do, but also to manage this division within the broader department. I'm deeply thankful for that. I'm also under no illusion that this level of commitment and this level of performance does not come without some sacrifice at home. And for that reason, I'd like to echo your thanks to Philippe, to Katleya, Richard, and maybe even Angelo for those times where Anabella is not available or work is occupying her mind to such an extent that she might not be there for you in the way that you had hoped. Thank you very much for granting us that time. Deeply appreciated. Finally, an evening like this does not come together by itself, so thank you to Professor Themba Mosia for his kind words and reflecting on the importance of an inaugural address within an academic's lifetime. Thank you very much to Professor Sanushka Naidu, the Head of Department of Biochemistry, Genetics and Microbiology, for joining us this evening. And then particular thank you to Marie Venter and the team from the Faculty Communications and Marketing for making this event happen. Again, thanks to Spatial Studios for professional service and the studio and the equipment and making it all happen. And finally, I'd like to end the evening with a message to vaccinate. It's the only way in which we can get back to something approximating normal, and I urge you to go and do that as soon as you can if you are not already vaccinated. Travel safely wherever you are, and good evening.